My pleasure now to have our invited guest lecture, final one of the day, Improving Diagnosis and Management of People with Brain Infections in Brazil, India and Malawi. Dr. Bhagteshwar Singh, a colleague of mine, we've worked together for a while now. It's a pleasure to have you here and to hear about this before we do a bit more of finishing up for the day. Bag.
Thank you very much, Ava, and everyone. Thank you for having me here. It's an honor to be invited to present today. But also thank you, I think, really for having me towards the end of the final session of the conference, because I don't think I need to explain all that much in terms of background, right? You're already sold, it seems, on the drive to do more research that improves care for people in low and middle income country settings. I feel like I've just been really well set up by the previous speakers. Although, those of you who aren't convinced, I don't know if maybe this would sway things. Maybe, if we were to do the vote again after this. No, sorry, I'm not trying to delay things further, sorry. Caught Ava's eye and thought, no. So I'm going to talk about the NIHR Global Health Research Group on brain infections, known as Brain Infections Global, shortened to BI Global. Some will call it BIG, but we kind of have avoided just being called the big study a little bit, because while it's big, we don't want to be reduced to just size, right? But a multifaceted intervention to improve diagnosis and early management of hospitalized patients with suspected acute brain infections in Brazil, India, and Malawi. That's it. That's all you need to know now. That's a lot of what I've said already, haven't I? But here's some declarations of interest, in case anyone is wondering, and a disclaimer. Professor Tom Solomon earlier mentioned that he was presenting something that isn't quite published, and things might change a little bit, and I just want to say, please don't go around telling everyone these are final results. It's still undergoing a second round of peer review at the Lancet, and so there may be subtle changes. I don't need to go through this slide, right? We all know that encephalitis and meningitis, there's been talks on meningitis today as well, is a really important issue throughout the world, and we've just heard that it's potentially we need to focus more on brain infections in places where more of them happen, in low and middle income country settings. But at the moment, we probably don't do enough on that. So in 2018, this group was set up, funded with £2 million from the UK National Institute for Health and Care Research. In fact, sorry, NIHR, the logo bottom right is actually out of date. Care should be in there as well. There were 5 interrelated themes that cut across each other to try and improve diagnosis of brain infections. And as you can see, we worked in these 3 countries. There were a variety of institutions involved. Cutting across these 5 themes was really important input from our public and patient involvement panel, which was chaired by Ava, and a big shout out to Encephalitis International for supporting that. There was representation from each of the 3 countries we were working in, as well as in the UK, where PPI has been embedded within research for much longer. And it was coordinated by the University of Liverpool, and just crediting the leads of the program there at the bottom. So we aim to improve diagnosis and management. I'm not going to say all of this again. Here are the aims on the screen, which I mentioned earlier, except to say that we were focusing on hospitals and wanted to try and cover tertiary and secondary care, and not just focus on specialist or central settings, which previous research mostly has. And we wanted to put together an intervention primarily aimed at improving diagnosis that was applicable everywhere and had some sort of structure, but was also sustainable and tailored to individual contexts. So this is a very simple overview of how we conducted the study. There was a pre-intervention phase where we charted out what was currently happening in each hospital in each country, what the challenges were to making a diagnosis, and what the capacity was or opportunities were for improvement. We then worked with hospital stakeholders of various sorts and policymakers beyond the hospital as well to co-design an intervention and then implement it in each place. and I'll show you a bit more of the process of that. And then we try to assess the impact of that. And alongside that, as many of you will know, when trying to improve quality of care, for example, in hospitals, it's always an iterative process, and you figure out things as you go along, what is working really well and maybe might inspire other hospitals versus what isn't quite working initially and might need some tweaks. So the design was a before versus after study, and I'll show you a bit more about how that played out. Overall, we recruited over nearly 2 years. There were 13 hospitals in our 4 centers in the 3 countries. You can see there's a wide spread of types of hospital. We were trying to cover different scenarios in each country as far as possible. And we estimated that so one of the primary outcomes, microbiological diagnosis, from the literature, we thought that it was a reasonable estimate to assume that 25% of people achieve a microbiological diagnosis, so finding a confirmed or probable pathogen that is causing a presentation with possible brain infection. And that a reasonable, meaningful, but also hopefully achievable point to bring that up to would be 45%. And accordingly, we wanted 900 patients to be recruited in each of the 4 centers, so 3,600 patients overall. And we analyzed using multivariable logistic regression with a range of predefined covariates within those models, and also with interrupted time series analysis, which I'll come back to. So inclusion criteria, these were generated for the purpose of the study and then validated using previous data sets within each country and another middle-income country. And as you can see, they were targeted at the front door of the hospital. So quite different maybe from the International Encephalitis Consortium criteria that we heard about in more detail earlier today. These didn't rely on test results and therefore availability of tests, and could be applied at the front door. And that's where we really wanted to target our intervention within the first day of presentation to the hospital. Our exclusion criteria essentially reflected the fact that we wanted to recruit adults and children, but neonates, as many of you know, are affected by pathogens that might be very different from infants and older children, and similarly with neurosurgical infections, the approach is very different. So the primary outcomes, and we had 2 co-primary outcomes of the study, which I can go into more detail how that amends things later. But the first was to make a syndromic diagnosis. So when someone comes to hospital, can we say with confidence that they have encephalitis or meningitis or another brain infection syndrome? Can we confirm to a point where that is what we are dealing with? Versus not really knowing that. And that could be something else as well. It didn't need to be a brain infection. We knew that we were going to capture a lot of patients who didn't necessarily all have a brain infection. And then microbiological diagnosis, which I mentioned earlier, which is finding the pathogen we think is causative. And then there were a variety of secondary outcomes, some of which were kind of process measures, seeing whether those kind of interim steps are being made in the course of trying to make a diagnosis. And some were really kind of patient-important outcomes like mortality, length of stay in hospital, quality of life, and neurological outcomes or function. This is how the study played out. We took a pragmatic approach to deploying the study and then the intervention when we were ready in each place. And you can see the red boxes with the smaller green bits within them are where we are studying what is going on and generating the pre-intervention estimates. The green within that overall red bar is really intense observation, and I'll come back to what that involved. And then the blue without any red at the far right is the intervention being in place. The red and blue crossover was where we were developing the intervention, but we hadn't yet implemented it anywhere. So that is, showing you how long it took to develop things, but doesn't include actual implementation. So when someone presents to hospital with a possible brain infection, what determines whether they get a diagnosis or not? And we thought about this as this is a causal diagram, or perhaps trying to generate a map of the processes involved. And you can see there are lots of processes involved. And many of them are influenced by how long it takes to achieve those processes or start them or kind of complete them, but some are time dependent, and some are dependent on resources, and some of those resources are kind of not really physical resources that are so easy to measure necessarily. So it's complicated, essentially, how you get from presenting to hospital to getting a diagnosis, and obviously we know of many complex scenarios where you have one simple intervention that cuts right through and makes a massive difference everywhere for everyone. But that's not often the case, and actually a lot of our time is spent, isn't it, thinking about how, earlier we were talking about implementation science, or how to deploy things, how things work in an interrelated way, and often the way then to deal with such problems is to try and tackle them from multiple angles. And so the UK MRC talks about complex interventions, and we followed their guidance on this to try and put something together that would do that. There are various ways of doing this. Just to take football analogies, I think our Danish colleague earlier was talking about how he would really relate to this, right? And you've got the idea of everyone being in the right place and knowing exactly what they're about to do, everything being detailed like a recipe, where you have not only the ingredients, but you also have do this at this point, that at that point, also watch a video. And then you have okay, here you go, we're not far from the goal, there's the goal, just get it in. And that's your brief. Don't care how you do it. Obviously, we went for something in between. Right, this is why I'm presenting them as diametrically opposite approaches. And what was really, really important, we felt, though, was tailoring to the context in each country and in each hospital. So we did some really detailed observation of the journeys of patients and their cerebrospinal fluid samples through hospitals to diagnosis, or often discharge without a diagnosis, or unfortunately sometimes death as well. And this is just a visual representation which we used in the course of the study to try and figure out what was going on at each point, with some ethnographic observation and a framework approach to analysis, not too far off actually the narrative approach that our colleague mentioned earlier. We also went into each laboratory and found out what was going on and what could potentially be leveraged to improve things in terms of brain infection diagnosis. And we came up with an overall structure or framework that we were going to try and follow. So at the top left, you can see that. We tried to be as simple as possible and capture everything within the system, drawing from the WHO building blocks of health systems that some of you will be familiar with. Then I mentioned earlier that we made sure we co-designed the intervention with people who matter. And that not only was led by centre investigators, obviously with people who were on the ground and responsible for working out what would be feasible and sustainable, but also there was oversight from a core working group trying to align things across all centres, and close liaison with our patient and public involvement panel to make sure that what we were doing was relevant. And a vital part of this was the involvement of policymakers from the outset at local, district and state or regional and also national level in each of the 3 countries. And so we had a policy working group that essentially didn't just advise, but helped build this intervention to try and make sure it would be taken up and scaled up later. This was the intervention. Well, I'm not going to go through all of this. A lot of writing here. I'm sure the slides will be shared later. And it says 10 here. Do I only have 5? Okay. Just some visual tools to show you what was going on here. Here is an example of a clinical algorithm, Vellore Center in India for children. Here was page 2 of a clinical algorithm in Brazil, which it's in Portuguese, so definitely not putting it up so that everyone can read everything. And here's an idea of what we were doing in terms of lumbar puncture packs and the guidance for them, which sometimes was where they were already in place, we were modifying them, and sometimes that was putting them in place afresh. Then we had a pathogen detection panel, very complicated, but essentially common and treatable tests initially that would be easily deployable. And then if those were all negative, then we went to more complicated tests for rarer pathogens. And then we had to train, obviously, people in laboratories and clinical environments. So in terms of patients, most of the patients, well, the majority of patients were adults, but about 40% were children. Most of them had an element of altered mental state or encephalitis, whether with or without some meningeal irritation features. And the majority had fever and altered mental state. You can see that there was more recruitment in the Indian centers than in Brazil and in Malawi. Here were the pathogens. Lots going on this slide, but essentially most were bacteria, most were treatable, which follows, and not really any huge surprises there maybe in terms of commonality. So interrupted time series analysis, what's all that about? So we know with these interventions that when you put an intervention in place, you can have a noticeable increase in, say, the proportion being diagnosed immediately, or that can be slowly over time, or you can have a bit of both. And we tried to look at these underlying trends over time to see if we were actually improving things beyond that underlying trend of improvement, which happens in every system, especially if you observe it, something called the Hawthorne effect. What we found in terms of our primary outcomes was our syndromic diagnosis increased overall, this is across all centers, from 77% pre-intervention to 86% post-intervention. That was significant on the multivariable analysis and also on testing the slope here. That was a significantly higher gradient of improvement or trend of improvement over time. Similarly, for microbiological diagnosis, from 22% to 30% was significant using the multivariable analysis. And we also had this slope, which looks a little less convincing, but is statistically significant post-intervention. So we had improvements in both of the co-primary outcomes and in a variety of secondary outcomes, and that was consistent across centers. We do have other elements of the study pending, and I can't show you results of those today. One thing I really want to say is that beyond the initial demonstrating that this can work, we also managed to improve things in a way that has sustained beyond the study, which was a key aim of what we wanted to do. And some examples, very briefly, we could make cancer diagnoses in Brazil that couldn't be made previously. We've handed over a lot of tests in each country to government-run labs who can then continue things. And rabies testing previously was only available at NIMHANS, one of our sites, and CMC Vellore can now do rabies testing. We're also supporting a strategy for the next 10 years for diagnostics in Malawi, which is really exciting and challenging, but exciting. Another thing we did was set up a network in order to investigate COVID-associated neurological disease, but that is now the Brain Infections Global Network, and those of you who are part of this will be invited to contribute to things that we're planning in due course. And training and capacity strengthening was really important at an individual level and in terms of the ability to deploy the NeuroID course in an adapted format twice now in each of these 3 countries, and webinars, and there are e-learning modules as well on our website, and I'll put our website up at the end. Here's a picture of NeuroID in 2020 in Malawi, just a few weeks before everything stopped. Next steps. So we're currently disseminating. This is part of that. Thank you for the opportunity to do that here today. There is as I mentioned earlier, a preprint online of a manuscript that hopefully will be published soon, and a toolkit that contains not only all of the tools within the intervention, but also all of the tools we use to actually do the study so that it can be replicated wherever you are in the world. We've been really lucky to be able to feed into national policy through our policy working group. There's a national guidelines group in India focusing on brain infections at the moment, and we're feeding directly, again, it's such a privilege to do so, into WHO policy. And further work will extend this to other serious infection syndromes, hopefully. We'll maybe try and leverage some of what we're doing for routine surveillance, and look at all-important outcomes, as pointed out by Ava earlier, to not just kind of look at what we're doing within the hospital and on discharge and very soon after. I just want to end by thanking everyone who's been involved with this. I think there are nearly 200 authors on this manuscript, so I haven't listed everyone here. But the patients and their caregivers need to come first, and obviously everyone who's been involved in the study, many of whom are actually here today or online. Thank you very much.
Thank you so much. Thank you, Bag. That's a fantastic overview. Sorry that we rushed you. It's that time of the day, isn't it? But we do have time for a couple of burning questions, if there are one or two questions in the please, before we do some of the exciting finish-up stuff, including prizes and so on. Do we have any questions? Yeah.
I won't be offended at all if there are none.
We've got one at the back there.
Thank you very much for the wonderful presentation. Just to confirm, was HIV an exclusion criteria in your study?
No, we actively included everyone apart from neurosurgical infections and other nosocomial infections and neonates. We included people with HIV and other immunocompromised. All right,
so I was just wondering, in terms of the etiology that you found, was it different from those who had HIV and the HIV-negative participants? Thanks.
Yes, for sure. And sorry, I didn't have enough time to go into much detail about the pathogens, but hopefully in the manuscript there'll be a bit more there. But yes, very different. As you'd expect, we saw a lot more cryptococcal meningitis, TB meningitis in patients with HIV infection, and some other things which obviously are opportunistic infections like JC virus or PML, etc. There were, this is an interesting point just briefly, that when we were constructing these pathogen panels, we did have separate panels for those with HIV, with known HIV from the outset or diagnosed during the admission, and for those without. And as you might expect, for common and treatable pathogens, we actually had quite a lot of overlap there. But I think finding different results kind of validates the fact that we did have separate panels there.
Hi there. Great stuff. Thinking about the children in particular, did you have any interventions that kind of addressed the complexities around consent? Because I know that in settings that I've collaborated with, there's often a degree of hesitation about it, association with bad outcomes, and often the person that can give consent isn't at the bedside, and I guess that's the premise for getting a lumbar puncture in the first place.
So consent for lumbar puncture? Yes, we trained people in performing lumbar punctures, but also within that was embedded how to gain consent and tricky scenarios where there'd be benefit from actual training and how to approach those. And we drew from previous work by colleagues, some of whom were in the room, to inform that. And that was something that actually the PPI panel were helpful with as well, in terms of maybe providing perspectives on how that could be approached.
Thank you very much, everybody. Thank you, Bhagteshwar, again. Thank you.